So far, we've talked about viruses and we've talked about prions, and these are non-living entities. Even though they mimic some of the characteristics that we associate with life, remember that they're things that cannot even reproduce on their own. They need help from actual living things. Today, we're going to start looking at living things and the basic building block of life. The basic unit of life is the cell. The so-called simplest cells are the prokaryotes, and I've put simplest in air quotes. I know you didn't see that, but even the simplest of organisms is still pretty darn complex. The prokaryotes exist as single cells, although they may sometimes link together in chains. They don't form anything more complex than that. Prokaryotes include the bacteria and also the archaea. You're seeing an example of a bacterium here. This is E. coli, which you are no doubt familiar with. You have a lot of these living in your body. Prokaryotes are defined by the fact that they do not have a nucleus. We have a nucleus in our cells. All multicellular organisms are made out of eukaryotic cells, and they possess a nucleus, and they possess organelles, the Golgi body, the endoplasmic reticulum, and so on, that we don't see in prokaryotes. And we'll talk about that in a bit. Prokaryotes were the first organisms, the first cells to appear on the planet, and they still really control what happens on the planet when it comes to life. They form the base of every food chain. In terms of biomass, they are by far more abundant than eukaryotic organisms. So biomass refers to the mass of living stuff, all the living goo on the planet. If you took all the giraffes and all the plants and all the, aardvarks, you took them all and you weighed them, measured all that mass of living stuff coating the earth, the biomass of prokaryotes is 10 times the biomass of eukaryotic organisms. They invented metabolism, and there's a number of metabolic pathways that exist in prokaryotes that don't exist in eukaryotes. They can get energy in weird ways that we can't. They maintain the nutrient cycles of the planet. So the reclamation, the recycling of sulfur and nitrogen, the breakdown of organic matter, the recycling of carbon dioxide, etc., all this stuff is being done in large part by the prokaryotes. And they're small. So what you're seeing on the left here is the head of a pin. Now a pin is pretty sharp, but seen very close up in this image that was taken by an electron microscope, you can see it appears kind of blunt at this scale. And what you're seeing there are clusters of bacteria. The cells are small, typically 1 to 2 micrometers in diameter. A micrometer is 1/1000th of a millimeter, so this is quite small. But despite their small size, they make up most of the biomass on the Earth. It's estimated that there are 5 times 10 to the 30 cells. That's a 5 with 30 zeros after it. That is a big number. Just in you, and this is an amazing thing to think about, there are more prokaryotic cells than there are eukaryotic cells. There are about 10 times as many prokaryotic cells in and on your body than there are your own cells. It makes you question what you are. Are you a human or are you an incubator? There are at least 6,000 agreed upon species. So these are species that scientists have said, okay, we can all agree that this set of characteristics, this shape, etc., that corresponds to a certain species like E. coli. But it's very difficult to identify separate species when it comes to prokaryotes. First of all, a lot of them look very, very similar. So just looking at them under a microscope, that's not enough. We can dye them with different dyes that will recognize different proteins and so on that are only found in some of them. That helps a bit. We can do some biochemical tests. We can see, what these individuals eat, what they can survive on. That might help us define species. But we can't use the classic biological species concept. If you remember, using that biological species model, we define species as populations that can't interbreed. They're different enough that they can't interbreed. Well, bacteria and archaea, they don't have sex. So that makes things difficult. And also what's really weird is some very, very different prokaryotes that are almost definitely different species can swap bits and pieces of DNA, and we'll talk about that later. So although we can agree on at least 6,000 species, there's lots more. And most biologists agree that there's probably millions of species. What you're seeing down the bottom here is an example of a rather common bacterium. This is Staphylococcus epidermis. And as we go through and look at a few bacteria species, pay attention to the names. They do tell you a little bit about the characteristics of that species. Staphylo, as we'll see, that refers to the growth form. These are bacteria that clump together. So individual cells that clump together, we call that arrangement Staphylo. Coccus means that they are spheres. It literally means in Latin berry. They're shaped like little round berries. So that refers to the shape of the cell. And the epidermis, well, that identifies where they live. This is a very common bacteria found on the surface of our skin. Prokaryotes have the generalized structure that we see here. They don't have a nucleus. They have DNA, of course. They have genetic material, and that genetic material is double-stranded DNA, just like we have in our cells, our eukaryotic cells. But instead of having multiple straight chromosomes contained within a nucleus, that's what we have, they have one massive circle of DNA. They have one chromosome. And it's not surrounded by membranes. It's not walled off in a nucleus. Instead, it's just free floating in the middle of the cell. We call that region where the DNA is hanging out the nucleoid region. They do have ribosomes, of course. Remember, what's going to happen is that DNA is going to be transcribed to make messenger RNA. The messenger RNA is going to be translated by ribosomes. And ribosomes are the smallest organelles. But they don't have other organelles. They don't have the Golgi body. They don't have the mitochondria, and we'll talk about why they don't. It's really quite fascinating. They don't have things like the smooth endoplasmic reticulum, the rough endoplasmic reticulum, etc. They are simpler than eukaryotic cells when it comes to structure. They have a cell wall. They have a rigid cell wall around the outside of the cell, so outside of their plasma membrane. And then in some cases, for some bacteria, they might have what's called a capsule, which is a thicker layer of protein. They might have a slime layer, which is a really gooey layer of proteins. Those proteins can help them, so help them survive harsh conditions, protect them from the environment in your body as well. So some bacteria will have capsules and slime layers that protect them from your immune system. They may have little bits that poke out that we'll talk about in a moment, and some of them have flagella. And the flagella, interestingly, spin. They spin like a propeller. That's interesting because in eukaryotes, the cells that make up our body, if there are flagella or cilia, which are the same thing but smaller, they don't spin. They wave from side to side. We talked about taxonomy earlier in the course, and if you recall, I mentioned that there were 3 domains of life. Domains are the biggest groupings of living things. So we have the eukaryotes. That would include us. We are made out of eukaryotic cells that have a nucleus and have other large organelles. Then we have the archaea and the bacteria. The archaea and the bacteria together can be referred to as prokaryotes. It's not an official taxonomic group. It's not a clade, as we talked about before. It's based solely on their structure, mainly the fact that they do not have a nucleus. The archaea appeared first. Archae means old. They are old cells. They were the first cells to appear on the planet. The bacteria appeared soon after, and the eukaryotes, as we'll discuss, are kind of a mixture of the two. The very first cells to appear on the Earth, the first archaean cells, were heterotrophs. Now, what does that mean? Troph means to eat. Hetero means something different. What it means really is they're getting their food from the outside world. They're taking it in. They're breaking it down. That's what we do. We are heterotrophs. So we take in food, and there is potential energy trapped within the bonds of the food, and we are liberating that. Later on, some cells evolved the processes needed to make their own food. We refer to those cells as autotrophs. Auto means self, self-feeding. So for instance, cells that are photosynthetic, they can make their own lunch. They can capture energy from the sun and use that to build complex molecules. So the sunlight is their source of energy. But prokaryotes took all the big steps. They invented these different metabolic pathways. For heterotrophs, things that need to break down outside food, the very first step is something called glycolysis. So this is a set of chemical reactions that occur within the cell. It occurs within the cytoplasm of the cell. And the very first cells developed this. And all living cells today, including our cells, are capable of glycolysis. With one really weird exception that I might have talked about before, chlamydia. We'll get to that. That's a really weird bacterium that has given up on glycolysis. But all other living cells that we know undergo glycolysis, and this is a process where they break down food that they either took in or made themselves and liberate some energy. Photosynthesis is a process that's absolutely essential to life on the planet, and it appeared in the prokaryotes as well. There are a couple other metabolic pathways that we won't get into too much in this course. If you are interested, I do have other videos on that you can watch, but it's not the subject of this course. But the Krebs cycle or the citric acid cycle, the electron transport chain, these are pathways that occur within the mitochondria of our cells, and they occur within aerobic bacteria. Those are bacteria that like oxygen. They can use oxygen. The Krebs cycle and the electron transport chain generate a lot more additional energy from our food, more than we can get from glycolysis alone. And again, all of these processes appeared in prokaryotes. The earliest fossils, so the earliest concrete physical evidence of life, dates back to at least 3.5 billion years ago. Remember, it's thought that the very first life appeared not that long after the crust of the Earth cooled, and it's thought that that first life must have been heterotrophic. It consisted of very simple cells that were wandering around in this primordial soup and gobbling up those self-replicating RNA molecules and any other organic molecules, proteins, lipids, etc., that might have been in the early oceans. It was gobbling that stuff up and feeding on it. The first really good fossils, though, come from photosynthetic organisms. They come from a type of bacteria known as cyanobacteria. And you're seeing images of those fossils here that date back to at least 3.5 billion years ago. We have cyanobacteria alive today that haven't changed very much. In fact, you can go out and look in your local pond and you can find organisms that look very similar to what you're seeing here. Here's some examples of living cyanobacteria. We've got 3 species here. They're all very common species. They're global in distribution, so anywhere in the world you can find these. If you wander down to your local lake or pond and scrape some goo off of a rock or a log, put that on a slide, pop that under a microscope, you're bound to see something similar to what we have here. On the bottom left, we have something called Glyocapsa. Now, a lot of cyanobacteria, they live as individual isolated cells, but Glyocapsa will divide and then the cells will remain in contact with each other and they'll glue themselves together with kind of a layer of like a jello gooey substance. And that's what you're seeing here. Keeping the cells together offers a bit of protection. It also means that the cells can share some excess food. They're making their food through photosynthesis, remember. On the right, we have another example. This is something known as Oscillatoria, another really common group of cyanobacteria. You find these in ponds and lakes. It's not one cell, it's a whole bunch of cells. So if you look carefully, you can see there's almost like segments. Those are individual cells. What's kind of interesting about this one, if you do have a chance to look at it under a microscope, is it waves back and forth. It's called oscillatorium or oscillatoria because it oscillates back and forth. And it does that to keep water moving over the surface of the cell so it can get rid of waste and it can take up lots of carbon dioxide and use that to make sugars and food. On the upper right, we have another cyanobacteria that grows as a chain of cells, and there are some specialized cells. There are cells known as heterocysts, and these are cells that capture nitrogen that's dissolved within the water, and they use that nitrogen to make ammonia and other compounds that the rest of the cells can use as a source of nitrogen to make nucleic acids and make amino acids, etc. So we've got a lot going on in these cells. They're making their own food. There's even a tiny bit of specialization in some cases. We have different cells with different functions, different roles. Not full-on multicellularity yet, where we have very, very well-defined separate tissues. We'll get to that when we talk about eukaryotes. But you can see life has already kind of got its act together within the cyanobacteria. Let's pretend we have a time machine and we go back about 3 billion years, give or take a few 100 million years, and we step out. We would instantly die because there's not enough oxygen. But let's say we were smart enough to put on a respiratory mask of some sort. This is what we might see. So this is an artist's rendition of what the Earth might have looked like about 3 billion years ago. We have volcanoes, we have some stuff going on, lots of geology going on. Not a whole lot in the way of life just yet, at least not something we'd be all that interested in probably as biased humans. The most advanced life at this point are these little rocky pillow-like structures that you see here. These are something known as stromatolites, and what they are is a bunch of cyanobacteria, a mat of cyanobacteria that has captured a lot of sediment and has become almost rock-like. So a stromatolite, kind of a pillow-shaped structure. It consists of these photosynthetic organisms, cyanobacteria, living in this kind of twisted, gooey state and trapping sediment, trapping sand and silt to create these hard structures. These stromatolites are still around, but they're not nearly as common as they once were. So back 3 billion years ago, they probably covered the shorelines over most of the planet. Nowadays, we only find them in a few isolated places. This is Shark Bay, Australia, so it's at the northern tip of Australia, and of course, the northern part of Australia is closer to the equator, nice and warm. This is one of the few areas of the planet where we still have these stromatolites. And another view of Shark Bay, Australia. What's happening is the tide comes in and it brings with it fine silt and clay and sand, and that sticks to the cyanobacteria. We would start out with just a thin film of cyanobacteria. It's producing these gooey, sticky substances to protect itself. The sediment sticks to that, and then the cyanobacteria tries to grow up through that because, of course, it wants to be at the surface so it has access to the sunlight. This continues over and over and over again, and if we were to take a section through one of these really, really weird rock pillows, what we would see is alternating layers of cyanobacteria and sediment. We have fossil stromatolites in Canada, especially in Ontario. So if you go up the northern shore of Lake Superior, for instance, you can find fossilized stromatolites. Here you're seeing an image of a dried up creek bed in Ottawa, and you can see stromatolites. These pillow-like structures, again, they don't look like much, but this was a very important step in the history of life on the planet. These stromatolites containing photosynthetic organisms produced a lot of the oxygen that eventually allowed more complex multicellular life to evolve. The atmosphere of the Earth, when life first evolved, contained virtually no oxygen. And that was a good thing. Oxygen is rather destructive. Think of what happens when you set something on fire. If you have a big bonfire, there's light, there's heat. That is a very intense destructive reaction where the organic molecules, the molecules that contain carbon that make up the wood, are reacting very rapidly with oxygen to produce that fire. Oxygen oxidizes things. It tends to break things down. And life probably wouldn't have evolved in an atmosphere like we have today. It wouldn't have been possible. But photosynthesis produces oxygen as a byproduct. It's fascinating what happens. Plants, algae, etc., that are capable of photosynthesis, they take water, H2O, and they split it, and they release that oxygen. And that oxygen proved to be very, very useful in the evolution of life. If we take a look at complex organisms like ourselves, we consist of trillions of cells, and we have a high metabolic rate. Oxygen can help us break down food molecules a lot further than we could in the absence of oxygen. We can get more energy from our food, a lot more, and that made multicellular life possible. But it took a long time to get to those levels of oxygen that multicellular life required. In fact, it took about 2.5 billion years of simple, single-celled photosynthetic organisms spitting out oxygen. Where did all that oxygen go? Well, a lot of it went into basically rusting the planet. Iron, as you know, grabs onto oxygen. Unfortunately, the iron in the steel and the metal of our cars combines with oxygen. It makes iron oxide. There's lots of iron in the crust of the Earth, and it had to rust first. And that's what you're seeing here. You're seeing some sediment that was once at the bottom of the ocean. It's been lifted up through mountain building, and you're seeing layers of iron oxide, basically rust. So even though these simple photosynthetic organisms were spitting out lots of oxygen, a lot of it was just being soaked up by the crust and being used to rust iron until it started to actually accumulate in the atmosphere and bring about the evolution of new things. Alrighty, before we move on and we start to look at the different groups in more detail, a quick refresher about the taxonomy of life. We have our three domains. We have archaea, bacteria, and eukaryota. The archaea and the bacteria, we can group them into prokaryotes, and again, that's just based on structure. We talked about viruses and prions before. They are acellular. When we're dealing with biological terms, if you see A at the beginning of a word, quite often that's used as short form to mean without. A means without, so without cells. These are non-living things. Viruses and prions are not built out of cells, and all life is either single-celled organisms or built out of cells. So we have the eukaryotes. That includes kingdoms, plantae, fungi, animalia, and protista. And then we have domain archaea, domain bacteria. There are, of course, smaller subdivisions within those groups as well, and we'll look at some of the major ones. Here we have a nice overview, a phylogenetic tree of the major groups of life. Again, we have our domains. We have the eukarya, the eukaryotes, and then we have the archaea and the bacteria, and the archaea and bacteria can be grouped together as prokaryotes, again, just based on structure. But I want to point out that bacteria and archaea are quite different in terms of their biochemistry, in terms of how they do stuff. They look very similar, they have similar structures, but they are different. I know that the terminology in biology can be really confusing. Now, if you know a bit of Greek or Latin, and I don't expect you to, but if you know a bit of Greek or Latin, the terms do actually make sense. And let's just break down this term here. Karyo means kernel. Nucleus. It's the nucleus of the cell. It's the kernel at the middle of the cell. And EU, if you see that in a word, means true. True kernel, true nucleus. So eukaryotes have a nucleus. They have their DNA packaged into a nucleus. Pro means before. So the bacteria and the archaea appeared before the evolution of the nucleus. We can trace all living things back to one universal ancestor. So in the very beginnings of life on Earth, when the very first cells started to form, they were fighting it out, they were duking it out, they were eating each other, but it's thought there was a clear winner that gave rise to life as we see it today. And very important things like glycolysis, like other metabolic pathways appeared in that common ancestor, and that's why all the descendants have things like glycolysis. They've preserved that very, very important, very fundamental metabolic pathway. All living things today, they use double-stranded DNA as their genetic material because that was something that the universal ancestor had. Now note that archaea and bacteria appeared quite early on. The archaea, if you note, are grouped a bit more closely with the eukarya. And that's because archaea and eukarya share a number of biochemical similarities that we don't see in bacteria. Now also there's some dotted lines there, and I'm not going to talk too much about this just yet. We're going to talk about this in detail when we talk about the appearance of eukaryotes. But what it's thought happened is that the eukaryotes evolved from archaea that incorporated bacteria into their cells. So we have these archaean cells that took up proteobacteria that developed into mitochondria. The mitochondria in your cells actually have their own DNA, and it's in a circle, which is something we see in bacteria and archaea. They have their own ribosomes. It's thought that your mitochondria are kind of captured, enslaved bacteria. And likewise, for photosynthetic eukaryotes, the chloroplasts that do photosynthesis are captured cyanobacteria that are now integral parts of the cell. Notice within the eukarya, we only have these three groups that have true multicellularity. The rest, everything else in that yellow bubble there that's not grouped within the multicellular organisms, are referred to as protista. And we will come back to that, so don't worry too much about that just yet. Now there are some exceptions. Seaweeds are included within protista, and they do have something that at least comes very, very close to multicellularity. So let's talk a little bit about the archaea. I am going to talk mostly about bacteria today because I think that's probably what most people are more interested in. Bacteria have more of a direct impact on our daily lives as humans. But of course, we don't want to ignore archaea because they did play a very important role in the evolution of life. And they are different from bacteria. Even though they're grouped together as prokaryotes, they are in their own domain for good reason. They do have some molecular structures and processes that are quite different from bacteria. On the right here, without getting into too much detail, we can see the phospholipids from bacteria and eukarya and from archaea. Phospholipids are used to build membranes. So the outer membrane of a cell, which is called the cell membrane or the plasma membrane, it is built of phospholipids and some proteins as well. Phospholipids are really quite important for forming compartments in eukarya. So the Golgi body, the endoplasmic reticulum as well, they're built out of phospholipid membranes. And you can see that the archaea have these little side chains on their fatty acids. So fatty acids are just long chains of carbon and hydrogen. They make up part of the phospholipid molecule, and they're branched in archaea, and they're not in bacteria and eukarya. This is a fundamental molecule that's used to build cells, so that's pretty important. Down the bottom there, you can see that in membranes, we have two layers of phospholipid. You can see the diagram there for the phospholipid bilayer from bacteria and eukarya. If you've done any biology, I'm sure you've looked at this in high school, you know that that's how most cell membranes, the ones that you've been told about, are built. But in archaea, we can have these long chains of fatty acids that stretch across the entire membrane, which is weird. Archaea also have these anchoring structures that we don't see in other cells, and they can use those to grab onto each other and cluster together for safety and support. One thing that sets the archaea apart from most bacteria and most eukaryotes is they're really badass when it comes to surviving really nasty conditions. So archaea appeared first. They appeared when the planet was not very hospitable for life. There was lots of incoming ultraviolet radiation because there was no ozone layer yet. There were these rather noxious, toxic substances in the oceans. And many of the archaea are known as extremophiles. Phile means to love. They love extreme conditions. That's what they evolved into. They're good at dealing with extreme stuff. We have some that are thermophiles. That means they love high temperatures. If you've ever gone to Yosemite Park, you've seen these hot springs. And by the way, if you haven't gone, definitely go. It is worth it. These hot springs are hot water geysers, and the water's coming out near boiling. And that's what you're seeing on the right here. So we have a pool of water that's almost boiling, and as the water moves to the edge of the pool, it starts to cool. And you're seeing all these different colors, blue and green and yellow and red and orange. That's different species of archaea that have different colors that are adapted to different temperature ranges, which is really amazing. So you have these really awesome colors. If you look at the upper right, we've got halophiles. Halo means salt. So there are some archaea that can do really well in very, very salty conditions. Most cells don't like salt. It draws the water out by osmosis. They dry up and they die. But there are archaean cells that can deal with that. So here we have a floodplain. It's being flooded by the tide, intentionally in this case. So you can allow the tide to come in to these pools and then wall them off and then allow the water to evaporate. That's one way that you can collect salt, sea salt. And we're seeing pools here of different salinity, and different species of archaea are adapted to these different levels of salinity. And we've got one pool there that's orange, one that's green, one that's red. Again, different species of archaea that are capable of dealing with different levels of salt. Down the bottom right, we've got a pool here which would be toxic to almost all living things. It's just full of iron, too much iron. So iron can be a good thing, but if there's too much, it does become toxic, and these cells can survive in that. In fact, some archaean cells are really good at extracting minerals. They actually pull minerals out of rocks and then release them into the water. So some archaea will produce these rather toxic conditions. Here's another example. The yellow streak that we see here is archaean cells that are very well adapted to high mineral levels. And we quite often see archaean cells like this growing in the tailing ponds from mining operations and so on. So toxic substances that would kill almost anything else. Some archaean cells actually thrive on that stuff. There are a number of archaea that produce methane as a byproduct, a waste product of their somewhat odd metabolism. And these are known as methanogens. Gen refers to genesis, to create, so they create methane. If you've ever put on a pair of hip waders and walked around in a marsh, you know that you release all this gas. All this gas comes up from the mud that you're stepping in, and some of it smells pretty bad. Those are gases produced by archaean cells. Methane, there's some sulfur gases, etc. And I should mention that archaea, because they appeared very early in the Earth's history, they evolved in an anaerobic environment. Anaerobic means without oxygen. And most archaean cells are poisoned by oxygen. Oxygen's very reactive. It kills them. So they live in environments where there's no oxygen. We refer to that as an anaerobic environment. And there's no oxygen deep in the muds and silts and so on at the bottom of a marsh. There are archaean cells that live in the guts of cattle, and they produce methane. You've probably heard that cow burps are bad for the planet because methane is a greenhouse gas, for instance. So I should mention that archaea are very cool and not harmful. They're not pathogenic. There are no known pathogenic archaeans, but some of them do produce some byproducts that can be harmful in some cases. I don't know about you guys, but I am a big astronomy fan. We live in a rather exciting time. It's quite probable that humans will land on Mars during our lifetime, maybe even in the next 15 years or so. Mars is really exciting for biologists because there might be life there. Now, it's going to be pretty simple life. It's going to be single-celled organisms living in the Martian soil. But that's a pretty big deal if we find life on another planet. If we do find life, it's likely to be something very similar to the archaea. In fact, it might even be archaean life. It's thought that archaea can survive the vacuum of space. So it's possible that Earth may have actually seeded Mars with life. But regardless, this is definitely something to keep your eye on. Will we find life on Mars? Will we find life on other planets in our solar system? Now, I'm not going to give my full opinion on Elon Musk, because that might be a bit inappropriate. But as you might know, he runs a company known as SpaceX, and they've developed this massive Starship, it's called, that they hope to use to carry equipment and people to the Martian surface. Now, this is something you might not have thought of before. We need to be careful not to contaminate Mars, especially with archaea from Earth. So if this Starship were to pick up archaean cells while it's being built, while it's flying through the atmosphere of Earth, there's a decent chance that those cells will survive the trip to Mars, and the cells may colonize Mars and contaminate Mars. Other space companies, NASA, a lot of international space companies, they take great precautions to make sure that their spacecraft are sterile. So they will sterilize them on Earth, they'll package them into a capsule that's sterile, and then they're not released from that capsule until they escape the Earth's atmosphere, for this very reason. And I don't know if Elon is taking the same precautions. I hope he is. I've searched, I can't find anything to suggest that he is. But a spaceship this size would be pretty much impossible to sterilize. So hopefully we don't contaminate Mars with life from Earth. We don't introduce foreign species before biologists get up there and have a chance to fully study Mars and document any life that might be there. Archaea are very well adapted to harsh conditions. And there are many other places in the solar system that it's possible we might find something like archaea. Venus, for instance, sometimes considered the sister planet of the Earth, is a very harsh environment. So although it may have started out something like the Earth, there was a runaway greenhouse effect, and the surface of Venus is incredibly hot. There's also rain made up of sulfuric acid. It's a nasty place. Maybe there's archaea in the atmosphere of Venus, or maybe even on the surface. Titan is a moon of Saturn, and it has oceans of hydrocarbons. Very, very weird environment. Perhaps that's something that archaea could use. Archaea can do some pretty cool things. Maybe something evolved there that can use that stuff as food. Europa is quite interesting. So this is a moon of Jupiter. And it's got a very, very deep ocean with a very, very thick layer of ice on top of that ocean. That's the image on the right here, and those cracks are massive cracks in this ocean. Again, maybe something that archaea could survive in. Archaea and bacteria evolved in a very hostile environment. The original Earth's atmosphere, as mentioned, contained no oxygen. The very early self-replicating molecules and the first cells were very fragile, and they would have been destroyed by oxygen. But bacteria, specifically cyanobacteria, developed this process of photosynthesis, which meant they could make their own food. And oxygen built up in the atmosphere to a point where it was a potential problem for cells. It might destroy the cells and destroy the structures within the cell. At this point, the archaea and the bacteria kind of went their own separate ways. The archaea remained very successful in harsh environments where there typically was very little oxygen. So down in the mud at the bottom of a pond, or deep down in groundwater. And they also survived in areas like hot springs, areas where bacteria were sort of shying away from. So they didn't have to compete with bacteria because of that, because they picked these really, really nasty environments that they were well adapted for on the fringe of existence. Bacteria became resistant to oxygen. Not all of them. There are still quite a few that are anaerobic, that are poisoned by oxygen, but some bacterial groups developed metabolic processes that could break down oxygen, get rid of excess harmful oxygen that might harm them, and some bacteria even developed a means of using oxygen to break down their food more completely, to break it down into carbon dioxide and water and extract every last little bit of energy. So for the rest of this topic, we'll talk about bacteria. Again, this is the basic structure of a prokaryotic cell. I think I've talked about all those structures. One thing I haven't mentioned is the plasmid. So in this drawing here, we can see a plasmid, and this is a loop of DNA. So DNA always occurs in loops in bacteria and archaea. Plasmids are much smaller loops of DNA, much smaller than the chromosome. So in a typical bacterial cell, we have one massive loop of DNA, that's the chromosome, and it's all twisted up. Imagine if you took a big elastic band and just twisted it. That's how the chromosome exists in a bacterial cell. Then there are much, much smaller circles of DNA. This is not to scale here. If it was, that plasmid would be too small to see, but the plasmids contain usually just a handful of genes. One or two, sometimes as many as 5, but those genes can be really important, and they can also be passed on to other bacteria. If you take a bacterium and you squash it, and you look at it under the electron microscope, you might see something like this. Imagine just taking one of these little cells and smashing it under your heel. So in this false color micrograph taken by an electron microscope, you can see the DNA that's spilled out from this squished cell. And if you follow it, it's actually all one piece. So this is one massive loop of DNA. Although bacteria do lack a nucleus, that chromosome does tend to be centered within the middle of the cell, and we refer to that area as the nucleoid region. Prokaryotes have small genomes. A genome is the entire set of information, the entire set of DNA within a cell. The reason that they have small genomes is in part because they're simpler than eukaryotic cells, for instance. They don't need as much information, but also they're incredibly efficient. In prokaryotes, typically all of the DNA codes for something. It all has a role. Whereas in eukaryotes, there's quite often a lot of junk DNA. There's bits and pieces of leftover genes that used to do something in our evolutionary past, but they don't anymore. There's bits and pieces of viruses that have become incorporated into our genomes, and there's a whole lot of DNA that we have no idea what it does or where it comes from. So in humans, for instance, about 90% of our DNA has no recognizable role. Prokaryotes, again, they're very efficient though. They don't have all that quote-unquote junk. We do have these much smaller circles, as mentioned, plasmids. They can be really important because they can contain genes that might, for instance, convey resistance to an antibiotic, and plasmids are quite easily exchanged between bacterial cells, as we'll see. Bacteria have ribosomes, of course, but the ribosomes are a bit different than the ribosomes in eukaryotes. They're smaller, and also they have a different composition. We can recognize the ribosomes as being different. They're made up of different proteins. They're made up of different sized pieces of RNA, etc. We quite often use the genes that code for the ribosomal RNA in classifying species. Despite the fact that plasmids are quite small, they're much, much smaller than the chromosome, and despite the fact that they don't contain very many genes, they can be very important. So as I mentioned, sometimes on plasmids we'll find genes that convey antibiotic resistance. But we might also find plasmids that contain genes that allow for the synthesis or construction of a pilus. A pilus is a hollow tube that can be used to transfer DNA from one cell to another. So let's imagine that we have a cell that has a plasmid that contains genes required for antibiotic resistance, and it also has a plasmid that contains genes that allow for the construction of a pilus. Well, now this bacterium can form a pilus, and it can transfer the plasmid that conveys antibiotic resistance to another cell, sometimes even a cell of a totally different species. And you can imagine how that might be important and how we might want to know that this is going on. In some cases, plasmids will contain genes that increase the virulence of a bacteria. They help the bacteria enter into other cells and cause damage, or they might contain genes that allow the bacterium to produce toxins or to produce their own antibiotics that they can use to fight off fungal cells, for instance. Or in some cases, there might be genes that will allow the bacterium to break down nutrients that it wouldn't normally be able to do. And again, the big deal here is that these genes that are found on plasmids can be transferred fairly easily to other cells, and then those other cells are going to derive these new characteristics. Now let's compare reproduction in eukaryotes and prokaryotes. Remember that in eukaryotes we have mitosis, which is asexual cellular division, and then we have meiosis, which produces gametes directly in animals. It's a bit more complicated in plants and fungi, as we discussed. But in eukaryotes, we have, of course, straight or linear chromosomes. We have several chromosomes. So in humans, we have 2 pairs of 23 chromosomes. The chromosomes have to be lined up in the middle of the cell along the metaphase plate. After the nucleus breaks down, they have to be segregated, the nucleus has to reform, etc. So this is a complex process which tends to take a bit of time. In prokaryotes, we don't have sexual reproduction. We have an asexual form of reproduction known as binary fission. Fission just means splitting. Think of atomic fission. You're splitting an atom. And what happens here is that we have our great big circular chromosome in the middle of the cell. It's going to divide during the DNA synthesis phase, and we're going to get 2 circles of DNA. Each of those circles will attach to the inside of the plasma membrane, and that's what you're seeing, that little loop that's sticking out the top. Now the cell will simply elongate, and that's going to pull those 2 loops of DNA apart. Incidentally, the plasmids will segregate separately. They'll divide, and then they will also move to opposite ends of the cell. One of the classic ways to identify different bacteria, at least it's a good starting point, is to look at the shape of the cells. Remember, the cells are very, very small, so they're 0.2 to 1 micrometers in width. And if we're dealing with rod-shaped bacteria, they might be 2 to 8 micrometers in length. We have 4 basic shapes. We have a coccus. That would be the singular, cocci if we're talking about several of these, but coccus means berry. We're looking at bacteria that have a spherical shape, so these are cocci bacteria here. We could talk about a bacterium being a bacillus. This is a rod shape, so it has one dimension, of course, that's longer than the others. It's kind of shaped like a hot dog. We could have Vibrio cells. These are shaped like a bent hot dog, like a banana, I suppose. And then we could have Spirillum bacteria, where the bacteria have a corkscrew shape. We can also identify bacteria based on the arrangements that they form. We can have diplococci bacteria. So again, cocci refers to the fact that they are spherical. Diplo means 2, so they tend to stick together in pairs. What happens is a cell will divide and it won't completely separate from the daughter cell. So we'll get 2 cells that are stuck together. If those cells divide again, then those cells will separate. We could have bacteria that grow in a chain, and that's referred to as the strepto condition. So a streptococcus bacteria consists of a chain of spherical cells. We can also have a Staphylo arrangement. This is where we have a cluster of cells. So they're not attached end to end in a chain, they're in a big mass. So in the top example there we have Staphylococci. Here we're dealing with cells that are spherical and they're stuck together in a mass. I should mention that we can have these arrangements with bacilli bacteria as well. So you can see in the bottom right, we've got a chain of bacilli. So that would be a streptobacillus growth form. So to summarize, we have coccus, which is a spherical shaped cell. We have a bacillus, which is a rod-shaped cell. We have a vibrio, which is a bent rod. And we have a spirillum, which is a wiggly corkscrew-shaped bacteria. We can have growth arrangements that are named diplo, where we have two cells stuck together, staphlo, where we have a mass of cells, and then strepto, where we have a chain of cells. If we were to continue trying to identify a species using classical means, after we had looked at the colony growth and identified the shape of the cells under the microscope and identified the growth formation or arrangement of the cells under the microscope, we might next look at the structure of the cell wall. So let's spend a bit of time talking about the basic structure of bacterial cell walls. They're made out of something called peptidoglycan. Peptidoglycan is a mixture of protein, that's the peptid part, and also sugar, that's the glycan part. You can see in the diagram on the left that we have two types of sugar, NAM and NAG. And I'm not going to have you worry about those names or the structure of these sugars. Realize they're somewhat similar to glucose. You can see on the right that we have chains of sugar. We have an alternation between these NAM and NAG sugars. Adjacent chains of sugar are held together by peptides. Peptides are short chains of amino acids. So I'll circle one. Here we have a peptide. You can see that it consists of 5 amino acids. Here you're seeing 2 of these peptides. These ones are a little bit shorter, but we've got these 2 peptides that are being held together by a bond. We've got a covalent bond shown in red that's holding these peptides together, and because the peptides are attached to these chains of NAM and NAG, those chains of sugars are also being held together. So we have these adjacent chains of sugar that are being cross-linked, held together by these short little proteins, these peptides. Here's another representation of the same thing. So we have cylinders that represent the chains of sugars, and then running at right angles to that, we have chains of amino acids that are holding those chains of sugars together. So it's kind of like a web of material. The cell wall of a bacterial cell is quite important to its structure. Typically, water rushes into a bacterial cell. We have pressure that builds up within the cell that's pushing out against the cell wall, and the cell wall is pushing back. We say that the cell is turgid, which means it's under pressure, and that's the normal, happy situation for a bacterial cell. You can imagine that if you weaken the cell wall, then there's a decent chance that the cell is going to rupture. There are quite a few antibiotics and other antimicrobial compounds that attack the cell wall for that very reason. If we can damage the cell wall of a bacterial cell, there's a pretty good chance it's not going to survive that. Two important categories of bacteria can be identified based solely on the structure of their cell wall. We can identify these different bacteria using a simple laboratory technique known as gram staining. Gram-positive bacteria have a cell wall that contains a very thick layer of peptidoglycan. That's shown on the left. You can see the peptidoglycan in purple, and notice that we have many, many layers of NAM and NAG sugars, held together, of course, by peptides. Underneath that, we have the plasma membrane. Remember, plasma membranes are made up of a lipid bilayer. So we have 2 layers of phospholipids, and then we have proteins embedded within that. There are other specialized proteins and also lipids and other compounds that are embedded within the peptidoglycan and also hold the peptidoglycan to the plasma membrane. Now let's compare this to a gram-negative cell. In a gram-negative cell, we have our plasma membrane, and then outside of that we have a rather thin layer of peptidoglycan. And then outside of that, we have a second membrane. Now don't get the two membranes confused. The plasma membrane is in direct contact with the cytosol. It's in contact with the inside of the cell. The outer membrane contains, again, some special lipids and special proteins that may give these cells some special properties. And once again, we do have lipoproteins and other compounds that are going to hold this structure together. Gram staining is a classic lab technique. It's been done for over 130 years and it continues to be important. After you've stained your cells, you look at them under the microscope, and if they stain pink, then we know we're dealing with a gram-negative bacterium. So this is a bacterium that has a plasma membrane, a thin layer of peptidoglycan, and then a second membrane outside of that. Cells that stain dark blue or purple are gram-positive cells, and those cells have a thick layer of peptidoglycan and only one bilipid layer. Here we're seeing those structures again. So in gram-positive cells, we have that thick layer of peptidoglycan. The rods are representing the sugars, and then we have peptides that are holding them together, and we have proteins and lipids that are holding that to the plasma membrane. In gram-negative cells, we have that thin layer of peptidoglycan sandwiched between two lipid bilayers. The space between the two lipid bilayers that contains that peptidoglycan is known as the periplasmic space, and it contains a matrix of proteins. It can be kind of gel-like. Within that periplasmic space, we may have some rather important enzymes that help to protect the cell, as we'll see. I mentioned earlier that bacteria have this weird little trick where they can transfer bits of information between them. Let's look at that in a bit more detail. Back in the 1950s, a researcher by the name of Joshua Lederberg did a rather interesting experiment. He had two strains of E. coli. So when we refer to something as a strain, we're talking about two populations of the same organism that differ in some way. So we had one lab strain of E. coli that was unable to synthesize a particular amino acid. He had another strain of E. coli that was also unable to synthesize one particular amino acid, but it was a different one. So just for the sake of simplicity, let's say that we have our one strain of E. coli here that can produce item B, whatever that is. It's something that's essential. It's an essential nutrient that bacteria would normally be able to make, but it can't make A, something that it should be able to make, but there's a mutation. It can't make that nutrient, and it can't survive without it. Then we have another population of E. coli, another strain, that are the opposite. They can make this A, but they can't make the B nutrient, and of course they can't survive without that. Now, if we were to explain the genetics of this, we would say that the first one is A negative, so the gene that's responsible for making the enzyme or whatever it is that's needed to make A is non-functional. Negative means non-functional, and the plus tells us that the genes that are needed to make that second nutrient are functional. Neither of these bacteria will grow on what's called minimal media. Minimal media is media that contains the bare essentials to keep a normal, healthy bacterium alive. So we have to supplement both of these strains. We have to give them the one thing that they're not capable of producing on their own if we want to keep these cultures going in the lab. But if we put them on, just the bare minimum that most bacteria need, they won't survive. Now, what he noted is that if he took these two bacteria and he tried to grow them on minimal media, they won't grow. We discussed why, because the first one can't make A and the second one can't make B, and they need that, and that's not in the media for them. But he found that if he took the two cultures and he mixed them together and then put them on a plate, he got growth. So how is that possible? Well, what he concluded, and it was later shown to be true, was that these two bacteria were swapping bits and pieces of DNA. This is a process known as transduction, where one cell picks up new information from its environment or from other cells. So what had happened was some of the cells that lacked the ability to make that A nutrient, whatever it was, they picked up the missing information, the missing genes that they needed to make the appropriate enzymes and so on from the other culture. And the reverse probably happened as well. But we ended up with some cells, not very many. So we've got billions of cells in these tubes, but we ended up with a few cells, maybe just a few hundred, that managed to get everything they needed. So obviously these bacteria were swapping bits of information. They were swapping bits of DNA, and that was a pretty exciting discovery. Lederberg went on to describe the process by which this occurs. So what we have happening is one cell producing a pilus, which is a structure that can transfer information to another cell. This structure is sometimes referred to as a sex pilus, as you can see here, but I would say that that's a misnomer. We don't really have sex occurring in this situation. Sex is where we have two individuals that come together to produce a brand new individual. So for instance, think of your parents having sex. I actually don't think about that, but your father and your mother each contributed a full set of information, a full genetic blueprint. Those blueprints came together in the zygote and produced you. And you, of course, are a brand new individual. Here, what's happening is information is being transferred from one organism to another. So think of this in human terms. If we could do this, what might we do? We might take our finger and poke the person next to us and transfer information to that person and change their eye color. I mean, that would be a pretty neat trick. So bacteria can transfer bits of information to other cells and change those cells, but they're not creating a new individual, so technically this is not sex. So what happens is this pilus is projected out and it grabs onto another cell, and the first thing that happens is it pulls that cell closer. The actual transfer that's going to occur is known as conjugation, but that very first step is pulling the two cells towards each other. And as the two cells are drawn closer together, the plasma membranes are going to fuse, and we're going to get this bridge or tunnel of cytoplasm that connects the two cells directly. Once this happens, the F plasmid in the donor cell is going to unravel. Remember, DNA consists of two strands. So one strand is going to be passed through that bridge over to the other cell. Once we have this single-stranded plasmid in each cell, that can serve as a template to make a new strand of DNA, and we end up with double-stranded plasmids again. Note that the donor cell is said to have a genotype of F positive. That means it has an F plasmid, whereas the recipient cell is F negative. It doesn't have an F plasmid, but at the end, both of these cells have an F plasmid. And now that recipient cell can make a pilus and share information with another cell. So this can spread rather quickly. Incidentally, I'll just point out that bacterial cells are haploid. They only have one set of information, and that's something that we may come back to. If we have a gram-negative cell that's placed in rather harsh conditions, what it will do is it will put up a fight. It has that outer membrane. It will do all that it can to destroy any toxins that are present, etc. Now, gram-positive cells have a different strategy. If gram-positive cells become stressed by a harsh environment, let's say we try to kill them with a bit of toxin or antibiotic or something like that. If they have enough time to respond, what they will do is they will form an endospore and wait it out. They will form this very, very tough, dormant, resistant spore that can survive very, very harsh conditions. And then when conditions get better, that endospore will germinate and give rise to a new active vegetative cell. Typically, only gram-positives make these endospores, but there are some exceptions to the rule. There are a few gram-negatives that can make spores, but that's rather rare. Anthrax is a gram-positive bacterium that can make these very, very resistant endospores. A lot of people don't realize this, but back in 2001, when we had the 9/11 attacks in New York, there were also a number of tainted letters that were sent to politicians. These letters contained anthrax spores. And in fact, several people were killed by these anthrax spores. You can see in the photograph on the right, we have Bacillus anthracis. This is what causes anthrax. And the arrows are picking out these very, very resistant endospores. Let's take a look at how endospores form in gram-positive bacteria. So we'll start with this cell here. Now, if this cell is happy, if there's plenty of water and plenty of food and the pH is right and so on, this cell will undergo vegetative growth. It'll divide by binary fission as quickly as it can and make lots and lots of little cells. But if conditions get harsh, let's say the water starts to dry out, the pH changes, maybe it's slowly being poisoned by some sort of toxin, then it will enter into this pathway. It will enter into a process known as sporulation. The cell is going to divide, but notice that the division is asymmetric. Instead of getting two cells that are the same size, we get one small cell and one larger cell. The smaller cell is known as the forespore, and the larger cell is known as the mother cell. Now something quite fascinating happens. The mother cell is going to engulf, it's going to eat the forespore. It's not going to break it down, but it's going to surround it. So you can see down here, we have one cell now, the forespore, that's entirely surrounded by the mother cell. And because of that, we have two membranes around that forespore, one that came from that smaller cell and one that came from the mother cell. Next, what's going to happen, that mother cell will continue to protect that cell. It's basically given it a big hug and surrounded it because things are getting bad out there. But the mother cell and also the spore are going to start dumping protein into that space between the two membranes. And we're going to build up this really tough cortex. The mother cell will keep producing more and more material as long as it can until it's killed by whatever the harsh conditions are. But by the time that mother cell is killed, we have this really, really tough, thick wall around that spore cell. The DNA breaks down within the mother cell, but the DNA is preserved within the spore. And that cell becomes dormant, and it can stay dormant for a very, very long time. But if conditions improve, then that spore will germinate and we'll go back to our starting cell. So this is a really fascinating way to survive harsh conditions. Instead of trying to deal with them, you just develop this really, really resistant spore. Endospores are remarkably resistant. In fact, endospores have been revived from vials that Louis Pasteur used about 160 years ago. So remember, he did those experiments disproving spontaneous generation. Well, a museum in France found some of his vials. They revived the endospores that were dried up on the inside of those vials, and they've been growing cultures of the same bacteria that Louis Pasteur used so many years ago. Endospores have been revived from Egyptian mummies, endospores that have been sitting in these dry conditions, completely dormant, for over 3,000 years. And this actually presents an interesting question. People that are working on these mummies are interested in how they died and also what sort of diseases and what kind of strains were going around at the time. And we've revived some strains that have been dead for a very long time. It's interesting because we can uncover perhaps plagues and outbreaks that killed people in the past, and we can study those organisms so that we can prevent it from happening again. But at the same time, there's the ethical consideration of should we be reviving these ancient strains? People have reliably germinated spores from Antarctic ice cores that are 8 million years old. So these are thick, thick ice layers that have built up from years and years and centuries and millennia of falling snow. That snow, that precipitation contained some spores, and they were buried in these very, very deep ice layers. Now, there are also some claims that endospores have been revived from the gut contents of a bee that was preserved in amber 25 million years ago. Amber is fossilized tree sap. So the idea is that this bee got stuck in some tree sap and became fossilized. Now this one I'm a bit critical of. There's some doubt around this one. And the reason for that is when you're trying to extract material from something this old, there's very little of the material. And if you're taking that material and you're trying to grow up endospores on a plate, it's actually more likely that there were endospores floating around in the lab that gave rise to these cultures. So it's a little uncertain. The other three, though, definitely they're well supported. So pili can be used to attach two cells to each other, and usually we're talking about one bacteria cell connecting to another for the purpose of transferring information, transferring DNA. We have these other appendages known as fimbriae, which are typically used by a bacterium to attach to a different cell type. So for instance, a bacterium might use fimbriae to stick itself to epithelial cells. Interestingly, quite often the genes that are needed to make fimbriae are also found on a plasmid, and they can be passed through a conjugation pilus. So in some cases, one cell that's all spiky, like the one in the lower left here, can pass that characteristic onto a cell that is naked, like the one at the top, and then that donor develops the ability to stick to epithelial cells and other cells. Here we're seeing fimbriae again, but we're also seeing flagella. Those are the longer appendages. Flagella are quite different in that they're not rigid structures that are used to attach to other things. Instead, they spin and they're used to move the bacterium. If an organism is capable of movement, we say that it's motile. Not all bacteria are motile. Some of them lack flagella. So for instance, almost all cocci bacteria lack flagella and are non-motile. In this diagram here, you're seeing a motile bacillus. So this is a rod-shaped bacteria that has lots of flagella. The flagella are going to spin, and you can see as they spin, they kind of get all tangled up and they spin together, and that causes the bacterium to move. Bacteria don't have a brain or anything resembling a nervous system, of course, because they are just a single, simple cell, but they can respond to gradients within their environment. So they can move towards something that they want to consume, and they can move away from threats. They can move away from toxins and so on. So they will move, and then they will throw out their flagella, and that stops them. It stops them in their tracks, and then they can change position and start spinning those flagella again. So they have this run and tumble kind of movement. In terms of both structure and function, the flagella of bacterial cells are very different from what we see in eukarya. So the flagella of eukaryotic cells wave back and forth, kind of like a fish's tail. Think of a sperm cell. We have a tail that goes back and forth. In bacteria, the flagella spins like a propeller. So we have this complex structure that is attached to essentially an electric motor. We have a structure that's anchored within the plasma membrane and also within the cell wall. In the diagram here, you're seeing a gram-positive cell. We have a thick layer of peptidoglycan that anchors the shaft of the base of this flagellum. Above that, we have a hook which acts like a swivel. Here you're seeing the same thing, but in a gram-negative cell. Notice that we have an outer membrane and we have a plasma membrane, and then in the space between the two, we have a thin layer of peptidoglycan. Embedded within that, we have our little electric motor, and once again, we have that hook structure that's going to act as a swivel. The photograph, or more properly, the transmission electron micrograph that you see in the bottom right, shows what this looks like under the electron microscope. Flagella allow bacteria to move around, and that's what you're seeing here with these E. coli swimming on a microscope slide. But how do we know that those flagella spin instead of waving back and forth? Imagine we have a bacterium, and let's say it's a rod-shaped bacterium, so it's a bacillus. And let's also say that it's a happy, active little bacterium. This bacterium is probably going to have at least one flagellum, depending on the species, of course, but let's say we've got a few flagella here. Now, what if we could attach one of those flagella to a slide? There is a way we can do that. So we've got our glass slide here. We can use antibodies. There are antibodies that will stick to the proteins that make up the bacterium. We can also make antibodies that will stick to proteins that we coat on the glass. These antibodies are going to act almost like a glue. So we can tether our bacterium to the slide. Now, if we're lucky and we get a few bacteria where we only tether one of the flagella, then when the flagella tries to move, of course, the cell is going to move instead because the bacterium can't go anywhere. Imagine that we had a situation where the flagella moved side to side, like the tail of a fish. That's something we would see in eukaryotic cells, sperm cells, for instance. If that were the case in bacteria, then when this little guy tries to swim, if we look at this under the microscope from above, what we're going to see is we're going to see the cell wave from side to side. But what if that flagellum spins? If that's the case, the entire cell is going to spin. Here we've got a bunch of bacteria, some of which we've managed to glue to the slide. We've tethered them to the slide by the end of the flagellum. Now let's see what happens if we let them try to escape. Notice that they're spinning around in circles. They're spinning around in circles because that flagellum is causing the entire cell to rotate because the cell can't go anywhere. The bacterial flagellum is a remarkable machine. What you're seeing here is a polytrichous bacterium, meaning it's got several flagella. We're looking at one and we can see how it's embedded within the cell wall, and this is the electric motor. There's channels there that hydrogen protons flow down and cause that motor to spin, which then causes the flagellum to spin. Spirochete bacteria are rather special. They have a corkscrew or twisted shape to them, and of course that would make them spirilla. They do have flagella, but they're not on the outside of the cell wall. These are gram-negative bacteria. They have a plasma membrane, and then they have an outer membrane. And the flagella are in between those two membranes. So for that reason, we call them endoflagella, endo meaning inside. The flagella are attached to an electric motor, at one end of the cell, and when this electric motor turns, it causes the entire cell to turn. So the cell will corkscrew through the water. When you're this small, water is rather viscous. It becomes almost like a gel at this scale. So this is a really interesting way of getting around. Rather than having free flagella that spin like propellers, the entire cell is going to corkscrew through the water. Here's a nice little animation that shows how this works. So again, we have these flagella shown in yellow here, and they're all bundled together. They're also called axial filaments. They have a motor at one end that's going to cause these long filaments, these long flagella, to spin. But because they're strapped to the side of the cell, because they're contained within those two membranes, they're going to cause the entire cell to rotate. As the cell rotates, because it has a corkscrew shape, it's going to push against the media. It's going to push against the water that surrounds it, and that will move it forward. What you're seeing here is one of these cells coming towards you. Spirochetes, as we talked about, have an unusual way of getting around. They have these axial filaments or endoflagella that are strapped to the side of the cell, and they spin the entire cell. An important member of this group is Borrelia, which causes Lyme disease. The Borrelia bacterium is transferred from one host to another by the tick. If you're bitten by an infected tick, the first symptom that you're likely to encounter is kind of a bullseye-like rash around the site of the bite. And it's important to get this treated quickly because the bacterium will spread into the bloodstream. It can become a systemic, body-wide infection and cause neurological damage. And the last bacterium we'll talk about today is Chlamydia, which causes chlamydia. These are tiny, tiny bacterial cells that live inside other cells and parasitize them. If you have chlamydia, then you will have these cells living within the cells that line the interior of your urogenital tract. As is the case with most parasites, they've evolved to become much simpler. So they evolve from more complex bacterial cells, but of course, if you're a parasite, a lot of the hard stuff, a lot of the work is being done for you by your host. So it makes sense to simplify. They've lost the ability to make ATP, which is really quite extraordinary. I can't think of any other cells that have lost that fundamental aspect of their metabolism. So they have to steal ATP from the cell that they're living in. They appear to be related to spirochetes, but because there's so little of them left, that's kind of open for debate. But they will enter into a cell and they will replicate within a vacuole inside the cell, and then that vacuole will ultimately rupture and release the cells into the interstitial space where they can go on and infect other cells. Again, there's lots of similarities with viruses here. That process of releasing the chlamydia causes damage to the cell, and also while you've got chlamydia inside the cell, the chlamydia is putting a huge strain on the resources of the cell. Here's a simplified phylogenetic tree for just the prokaryotes. You can see with the archaea, we have the extreme halophiles. They love salt. We have the methanogens. We have the hyperthermophiles. They live in water that can be at the point of boiling. So we talked about those groups. And then in bacteria, I've used some arrows to pick out the groups we just talked about. The gram-positive bacteria have a plasma membrane that's surrounded by a very thick layer of peptidoglycan. We can identify that by simple staining techniques in the lab. We have the proteobacteria, which is a really big group. It includes the bacteria that gave rise to mitochondria, We'll talk about that in our next topic. It includes E. coli and a whole bunch of other important bacterial species. They're gram-negative. That means that they have their plasma membrane, their cell membrane, and then they have a thin layer of peptidoglycan outside that, and then another membrane. That second membrane gives them two chances to filter what gets in and out of the cell. So both of those membranes are selectively permeable. What that means is the cell can exclude some things the bacteria doesn't want, and it can allow in things that it does want. So the cell has two chances to limit, let's say, the influx of toxins, for instance. So they do tend to be kind of hard to kill. We have chlamydia, which is a very weird one. Again, it's a very simplified bacterial cell. It's the smallest living cell that we know of, and it's given up on things as basic as glycolysis. It doesn't manufacture its own ATP. I'm not sure if I mentioned ATP, but that's a molecule that's used to trap energy. And it just steals that molecule from the cells that it lives in. It lives inside other cells. And we have the spirochetes, which are those weird corkscrew-shaped cells that spin through the water. And here again, just a summation of some of the big groups of bacteria. The proteobacteria, the biggest group, they're gram-negative. We have the chlamydia, all parasitic. We have spirochetes. Those are those weird corkscrew spinning cells. We have the gram-positive bacteria and the cyanobacteria. I would say the cyanobacteria are the most important because they make most of the life on Earth possible. If we did not have cyanobacteria, we, as humans, would definitely not be here. So cyanobacteria produce a lot of oxygen. That was really important in the early Earth's atmosphere. Still important today. Cyanobacteria form the basis of a lot of food webs in the ocean, in lakes, in ponds. And as we'll see next time, cyanobacteria gave rise to chloroplasts, the chloroplasts that are in plants and algae that also produce a lot of oxygen. There are pathogenic, so harmful, species found in all of these groups except for the cyanobacteria. Escherichia coli, E. coli, is a bacterium we'll spend a lot of time talking about and also working with in the lab. It's a nice example of a typical proteobacterium. It is typically helpful to us. It lives in the gut and it releases vitamins for us and it helps us break down our food a little bit more. But there can be strains that cause us problems. Escherichia coli is sometimes referred to as the bacterial lab rat. And what I mean by that is a lot of what we know about bacteria has come from the study of E. coli. We have sequenced the entire genome, and that's what you're seeing on the left there. And we use this bacteria to help us in other endeavors. We use it to amplify or make copies of bits of DNA from all sorts of things. We use it to manufacture pharmaceuticals as well. So we can take the gene for human insulin, take it out of a human, put it into a plasmid, put that into E. coli, and we can trick E. coli into producing human insulin, something, of course, it wouldn't otherwise do. I hope you've enjoyed our brief little introduction to the prokaryotes, the bacteria and the archaea. They are, of course, different from eukaryotic cells, and at this point you should be able to identify a few differences. We're going to talk about eukaryotes in our next topic, and we'll spend most of the rest of the course talking about eukaryotes. Prokaryotes are simpler than eukaryotes, but again, anything that's alive is still pretty darn complicated. Bacteria are classified based on their shape and their structure and their biochemistry, their metabolism, etc. But nowadays, it's usually done by DNA sequences, specifically DNA sequences, genes that code for the ribosomal RNA that we find in ribosomes. Bacteria can be named based on their shape. So now, hopefully, you can tell your friends what Staphylococcus means, for instance, or Streptococcus. The name identifies the shape of the cell and also how the cells are stuck together. Now that's not always the case. There's lots of names that are based on other things, other properties of the cells. And a few terms here for you. We have things like the nucleoid region. Again, that's where the DNA is found in a prokaryotic cell. It's not found in a nucleus. Peptidoglycan, that's what the cell wall is made out of. It forms one of the most important layers of the cell wall in most bacteria. We have flagella that move the cell around. Remember, a capsule is an outer coating that's found outside of the cell wall. Also a slime layer, that's a really loose, gooey layer that can be found outside the cell wall. And capsules and slime layers protect bacteria from their environment. If they're living inside you, it protects them from you. We have apilus, which is used to transfer information from one cell to the next. We'll talk about that a little bit more in our next topic as well. Endoflagella, those are flagella that are strapped to the side of the cell. We have a bunch of terms here that describe the shapes of different bacteria and their growth forms. And then the last one there, extremophile, remember that pertains mostly to archaea. These are cells that like things really difficult. They do well in very intense environments. And a few study questions here to round things out. All right, so next class, as mentioned, we're going to talk about eukaryotes. We'll talk about the big things that appeared in eukaryotes, like, for instance, sex.